Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm Peter Bannattini, and here I discuss brain imaging with scientists from around the world. Today, my guest is Dr. Angie Laird, who trained as an imaging physicist but has evolved into a cognitive neuroscientist and a true pioneer in meta-analysis of fMRI data. Angie has spent the bulk of her career developing novel data analysis algorithms, neuroscience informatics tools, and neuroimaging ontologies to yield analytic strategies for improving investigations into functional brain networks of healthy individuals, as well as in populations with psychiatric and neurologic diseases and disorders. Early on, she has seen the untapped value in meta-analysis and has fostered growth in this fundamentally important area in functional brain imaging. Dr. Laird received her BS in physics from Florida State University in 1998 and her Ph.D. in medical physics from the University of Wisconsin-Madison in 2002. She was a faculty member at the Research Imaging Institute of the University of Texas Health Science Center in San Antonio from 2004 to 2012. And currently, she is professor and director for imaging science at Florida International University in Miami. Along with her development of meta-analysis tools and her own research, she plays a central role in the Adolescent Brain Cognitive Development Consortium, or ABCD Consortium, which is the largest long-term study of brain development and child health in the United States. This was a great conversation that spanned the early culture of fMRI research, early efforts towards data sharing, to the current practices today where data sharing and analyzing data across studies and from large shared data sets is becoming the norm. We also spent time talking about the origin, logistics, and the impact of the ABCD project. I hope you enjoy it. Today, I'm here with Dr. Angela Laird. As you heard in the introduction, she's, I would say, sort of a unique pioneer in pioneering the area of not only open science, but also more specifically, meta-analysis. She's created a bunch of really useful tools, and she sort of increased everyone's awareness of the need for meta-analysis, how to do meta-analysis, and has been working also specifically on applications of meta-analysis, but also other things too, more sort of focused neuroscience studies as well. So, Angela, thanks for coming on.
Thank you so much for having me.
So why don't we just start? I always like to start with like a history, a little bit of background of how people get started and what was important in starting out. First, I mean, your initial bachelor's in science was in physics, but how did you sort of gravitate from that to brain imaging?
I'll probably back up just a little bit. I was a pre-med major for 4 years as an undergraduate student. I did not come from an academic background. I didn't come from a science background. And what do you do when you're smart? You have people tell you, oh, you should go to med school. But I switched my major after my very first physics course. I fell in love with actually using my brain in order to solve problems as opposed to just memorizing things. I joined a nuclear physics group, and I realized I wanted to pursue a career in research. So then I transitioned to the University of Wisconsin for graduate school, and I really wanted to join Peter Timby's cosmology group and search the stars for clues about the origin of the universe. But he wasn't taking on new students. And so like that little thing totally shifted my trajectory. I wandered across the street to the medical physics department. And that's where I met Beth Meyerand, who you know. Yeah. She was a brand new assistant professor that year, and she was looking for new students to take into her lab. And we immediately clicked. And, the rest is history. What's interesting is that I've always been drawn to solving puzzles from data. And so cosmology, that's what I was attracted to in there. But neuroimaging has offered me exactly the same thing. You're looking for really small signals, really important signals in a lot of noise and a lot of data. So yeah, that's how it happened.
Oh, that's great. That's great. So Beth took you on, and Beth had right. She actually wasn't necessarily in fMRI. I was a fellow graduate student with her, and it seems that she was more, did some great work in image reconstruction methods and sort of
Yep.
MR physics stuff. And so
Yep.
was your PhD in MR physics or is it?
So that was the tricky bit, right? Because I wasn't even a PhD in the medical physics department. I was a PhD in the physics department. So I had to find a way, physicists, they love their disciplinary boundaries. And so I had to find a way to make my committee and my department think that I was getting a physics PhD. And so I actually, the hook that we came up with was I applied chaos theory and time series analysis to fMRI time series, where I kind of blended those two. And that seemed to work. They gave me the diploma, so I'm good to go. But yeah, like that was actually a bit of a struggle for me in the beginning, because again, I love physics problem solving, but I kept being drawn into this mystery of the brain and how do we really solve that. And so when I finished up at Wisconsin, I took a postdoc at University of Texas Health Science Center in San Antonio. And being in a medical school was immediately more freeing for me research-wise, that it no longer became about like solving physics problems about the brain, but it just became about solving biomedical problems so that I could use my physics skill set, but not really be constrained in terms of the direction that I went. And so over time, I really sort of naturally evolved more into cognitive neuroscience.
That's okay. That's great. That's great. And actually, I mean, it's funny because it might come full circle in some sense because, the physics aspects of, I mean, there's many more physicists coming into the field of fMRI and sort of applying, trying to look at, I mean, like you said, looking at things from more of a first principle perspective.
Yes.
It's really trying to understand, the essence of how things work as opposed to descriptions or whatever. So yeah, that's actually, I remember I also, we overlap a little bit as well. I also considered postdoc with Peter Fox when I was looking around initially, and he was just getting started with 2 Tesla Elson scanner, I think they had. And so that's great. So, but with Peter though, it seems like your whole career took a wonderful turn in the sense where, you started, and he had his brain map database that he was one of the only groups that had this database with more it's a coordinate based database. And
Right.
there you were tied in with this whole community of people that's
Right. At that time, as hard as it is for folks of today to imagine, there wasn't, I want to say hardly any data sharing going on. And so meta-analysis was really the only pathway that we had to really look across studies, right? If you didn't have access to the raw data, if there were no open access data repositories, and super big nod to Jack Van Horn for the efforts that he led around this time. But the reality is that it was just a very limited opportunity to look at multiple data sets. And to look at open access data sets. And so meta-analysis was a way where you could dig into the literature and we could have a whole debate on the real value of coordinate-based meta-analysis as opposed to having actual raw MRI images. But at the end of the day, it was feasible. Yeah. If you had enough time, you could sit down with a whole bunch of PDFs, you could scrape those coordinate tables, and you could do something with them. And so for me, meta-analysis is just this beautiful opportunity to take a step back. There's so much fMRI research that's being conducted by amazing scientists. Today, it is overwhelming for our students to really look through the literature in any systematic way, especially if they're going paper by paper. So we really need meta-analysis to collectively synthesize what we know and where our gaps are. And again, for me, there's a lot of nuance into the meta-analytic approach and the algorithms, but at the end of the day, I am comforted by the idea that I am just trying to find the strongest signal from all of the noise in the literature. I think that there's a lot of meaning to that.
Definitely. And we'll get into that, but it's actually interesting that you bring up, there was the BrainMap database and also Jack Van Horn's
fMRI DC.
Yeah, fMRI DC at Dartmouth. Gazzaniga started that up. And that was actually, I remember being there when that started. And I thought that was just an amazing thing to start. And I think there was a lot of concerns. And I remember that Gazzaniga even, it took a heavy-handed approach initially. I think where he said, well, unless you're going to put your data in this database, you can't publish in
Journal of Cognitive Neuroscience.
Yeah. And I think there was a little bit of backlash to that. And I think that,
and if you talk to Jack, they would get the most passive aggressive submissions where they would just get like these manila envelopes shipped to them just full of data trash, right? Here you go, it's submitted, right? So, I mean, it was definite growing pains for the field during that era.
Yeah, and that's actually funny that you mentioned the mail, because the data was so large that
Yeah.
if you requested data, they mailed you a disk or something, and you were all thinking, how is this gonna work?
Well,
Um,
it did, but we needed that in order to move to the next phase, right?
yeah, that was really prescient as far as that's concerned. But and right, and Peter Fox was sort of way ahead of the curve as far as thinking in terms of making this database as well, and which was, and I think actually, and right, there are concerns. So and concerns are that we're still struggling with today, that we might as well just talk about briefly a little bit. There's, of course, I remember when I was talking to Jack about this a long time ago, thinking, okay, so how are we going to really use this data unless it's curated in some way, and how, and, unless there's some standards in how it's collected, in terms of the experiment or whatever, because it always seemed that data was, initially the goal was not to necessarily pool the data, but to reanalyze that data
Right,
set.
right,
And so that's a little bit lower bar in some
right.
regard, because nobody even thought like, okay, we're going to actually put this disparate data together in some way.
That actually was the early conversation about the multiverse. You know, if you analyze data in different ways, do you get different results?
And of course, with fMRI and with many other, but especially fMRI, I mean, you have differences of acquisition. You have different experimental protocols which look at many different things, from magnitude to latencies to undershoots to fluctuations, and then you have different, as you said, and now this is coming out all the more. This metaverse of processing methods that could all give slight and different thresholds, and nobody really agrees exactly on a threshold, and so we're still struggling with that to some degree. And there's different qualities of data as well. Before we get into specifically your tools, maybe a general question of that still seems to me like potentially an issue, but it seems like maybe it's overcome by just the sheer numbers of, in terms of doing the meta-analysis. It would just sort of, that would be part of the noise.
That's my perspective entirely.
Okay.
Is that, yeah, there is a lot of variability if you drill down into, our analytic design, our research design, how things were analyzed, these, like you said, specific p-values, specific whole approaches. But there's thousands of studies out there,
Yeah.
and I don't build tools for experts. I build tools for students.
Yeah.
I build tools for the 19-year-old me that was wandering around going, the brain? We look at the brain? Okay, let's do that, right? It is a very overwhelming barrier. The literature is an overwhelming barrier to folks who want to get into this field. And so, as a professor, for those who join my lab, their first step is, okay, what topic are you interested in? Let's do a meta analysis, and making sure that the tools are user friendly so that students can jump in. There's plenty of time later to teach them about all of the nuance of statistical analyses, all of the image reconstruction to the point where you're thresholding math. But what they need to have is a real solid foundation into what studies have been done in their specific domain already.
Yes.
And so they learn literature search skills, but then they also learn about putting a whole bunch of studies together and seeing what signals rise to the top.
No, and actually that's awesome. I think it's a nice on-ramp in some sense for students.
It's an excellent on ramp. It's great for experts too. I don't want to, push them to the side. And we have a whole lot of meta-analysis papers where you have somebody who's been deeply embedded in the field for 20, 30, 40 years. And it's a good opportunity for them to take a step back and say, okay, look, I have this idea that's really been bugging me. I'm going to drill into the data to see if this theory holds water. So meta-analysis is for everyone, but yeah, like you said, it is a fantastic on-ramp project for novice students.
We can talk a little bit about the tools, but one thing that just strikes me is that I've done that to some degree, like, just for the fun of it, with neuroscience, for instance. And, you could put in anything. You can put in any word, like a keyword, and it will come out with a map.
Yeah.
of like what's associated with that word. It's a nice place to start in some sense. So, you can sort of, but you always take things with a certain grain of salt that, obviously it probably misses a lot, or there's probably some nuance, but at least, generally the areas that seem like they show up with like schizophrenia. What are the areas that are sort of, or actually, no, it only shows not for I haven't looked at it recently. Is it for disorders or just for tasks themselves or behavior? I'm not sure, but
It was originally it was a bag of words approach, so it's for terms.
okay, just terms in general,
Yeah,
topics, right?
yeah.
So what's the difference between, and I could do a set of studies. I could just iteratively go through neurosynth and find these areas, and they give you some sort of statistics associated with that and number of papers and whatever. And what's the difference between that and, for instance, the tool set that you've built with, like, for instance, Nightmare and
So I'll start with brain map
okay,
because that's where I started.
yeah,
So it was in 2003 that I started working on brain map, and at that point we were building the applications for users to interface with the database. Meanwhile, I was a brand new postdoc, and I do what postdocs do, which is tasks no one else wants to do. So I was sitting in a little dark cubicle manually annotating papers for 12 hours a day,
oh
right?
man,
So I was building the brain map database.
wow,
Um,
wow,
and so that was my first 6 months of my postdoc. And it gave me, as tedious and mind numbing as that was, it gave me a real bird's eye view, the landscape of the field, what folks were doing, and what we had learned so far.
yeah,
And so then we started having a conversation with Peter Turkeltaub, who was a graduate student at the time, I think under Gwen Eden at Georgetown. And he had just sort of casually invented the activation likelihood estimation AL meta analysis method because he needed a tool that would help him understand localization of pet data. We had conversations with him of like, hey, this thing looks like it could be really useful if combined with brain map data. So we started developing what's known as Ginger Ale, which is the meta-analysis application in brain map that combines the Ale approach with brain map data.
okay,
So I want to say that a lot of my perspective was formed in those early years, that the automated tools that came later, it wasn't something that I naturally gravitated towards or even originally understood how powerful it could be. I was really focused on students need to do lit searches, and lit search is this sort of glossy, lacks substance. Like, how do you show them how to do that, and what do they do with it? Is there a product associated with it? So meta-analysis became that product. Do your literature search, get those coordinates, and then bing bam boom, you have an image that represents convergence in that domain. And so for me, it really was about the manual labor. Like, computers were great in 2003, but nothing beat the intelligence of a human being sitting in a dark cubicle going through the literature paper by
yeah,
paper. And so I still like for students to put in the effort to really get their hands dirty and to look at each paper and read each paper. That's when you're dealing with a small set of the literature where maybe there's 20, maybe there's 50, maybe there's 200 papers. Once you get larger than that, you get into the thousands, then yes, the automated tools are very, very, very valuable.
yeah,
So it was in 2011, I think, that Tao Yarkoni published the Neurosynth paper,
yep,
and we were all blown away. It's like, oh my gosh, I've been sitting in this cubicle literally for years, and boom, he's done this automated approach where he's just scraping the tables and building the database. And so for a number of years, it really was this lovely balance. People would say, well, should I use BrainMap or Neurosynth? It's like, okay, well, let's talk about what your research question is, because the tools, they do very, very different things. So then I left San Antonio in 2012, and I naturally started gravitating to other things. This is probably the longest that I've had a conversation with someone that the ABCD study doesn't come up. So that sort of took
talk
over my
about
life.
that, yeah,
Yeah, that took over my life in 2015. But during that transitional period, moving from San Antonio to, I'm here at Florida International University, FIU in Miami,
yeah,
I shifted away from the BrainMap project, but I got a some new faces into the lab. And I met Taylor Salo, who was a graduate student at my lab. And Taylor very much wanted to build a new meta-analytic platform that was really agnostic to this whole, well, is it brain map? Is it neurosynth? Is it this tool? Is it this database? Is it this algorithm? Like all of that was noise to Taylor, that he just wanted to say, let's level the playing field so that you can have one software package that can utilize different metadata from different databases, that can utilize different algorithms. BrainMap is very much an AL meta-analysis method platform, whereas Neurosynth has always been very much an MKDA developed by Tor Wager platform. And you never had people doing MKDA with BrainMap or AL with Neurosynth. And Taylor's Nightmare, he developed the name again with Tao Yarkoni, Neuroimaging Meta-Analysis Research Environment, that took away all of those barriers and boundaries. And so it really is just a Python package that says, okay, here are the opportunities to you. You get to build your own meta-analytic framework.
And so in the way that allowed you, let's say you were starting out with Nightmare then, and it would, so when you say building a meta-analytic framework, you basically, so it's you say it's agnostic to both of these, but does it use these in some ways, or does it, you just decide in some sense, right?
Yes. It uses Neurosynth data a bit more than BrainMap data. BrainMap data formats can be used, but BrainMap continues to be a closed source platform, whereas Neurosynth is fully open. Okay. So we do get a little bit more use of the Neurosynth platform within Nightmare.
So just a really basic question. So BrainMap still relies on coordinate based, and I think Neuroscience is more. They've encouraged unthreshold maps in some sense, and I can imagine, right? I mean that's one source. It seems that it seems like you would miss a lot with coordinate based if you have threshold set, and let's say there's some subtle effect that's very common. I think of the power of, like, for instance, multivariate imaging is where you have many things that are below threshold, but then that gives a pattern that gives you something. Whereas if you just rely on the coordinates, you would never see those, and they would never help boost things up in some regard. I mean, so it seems that, but of course, there's problems with working with unthreshold maps. I mean, you have to, there's different tortuosities of activation. There's different quality of the data itself is not as process in a sense where it's not as clean, I guess.
But that's okay. There's trade-offs to everything that we do. There's trade-offs to structural MRI to functional MRI with our whole added dimension of time, right? So for me, coordinate-based is, yes, more coarse, not as proficient in identifying, the intricate extensions in 3D space of all of the blobs that we're trying to look at and layer over on top of each other.
Yes.
But if we didn't have coordinate-based meta-analysis, there's huge chunks of the literature that we would not be able to access.
Yeah.
So, I feel like there's a time and a place for everybody. Yeah. Everybody gets to belong. And everybody gets to do their thing. And we take all of the information together and we try to find something, a meaningful way forward.
I love the idea of meta sort of trying to actually synthesize and bring together in some way, all this work. Because when you really think of literature and brain mapping in general, what do we have? I mean, it's basically, we look at a paper and we say, oh, okay, that's interesting information. And, it might be, where is the information actually kept in that sense? So if you start to synthesize things a little bit more, you can start to have at least a place where it's described. And so my question is, let's say, meta-analysis continues on, which it will, and grow. You'll have more and more, it's more and more data about descriptions of, different populations and ever more subtle, maybe differences. And so what's the goal? What's finally would you do with that? And so the reason why I'm saying this is because I'm always thinking about building sort of models of the brain, sort of, instead of having just a description and a really good one. I've often thought of trying to figure out ways of plugging this into some sort of model of, where you try to, really understand how the brain is functionally the principles of functional organization. So that's a place to start. But I mean, what are your thoughts? I'm going on.
I mean,
And
Peter, are you trying to tell me you want a computer that can basically function like a brain?
well, someday, but yeah,
That's a big step,
yeah, that's
right?
huge.
It's absolutely huge because it's not just one thing and it's not just a couple of things.
Right.
So I think our field, as fMRI has evolved, we've gotten progressively more and more aware that it's not just a brain in a scanner doing this one thing at this one moment in time, that our brains are this extraordinarily complex representation of all of our past experiences,
Yeah.
both genetic and environmental. And so there's a lot of focus on what's the next imaging modality. I'm like, I don't care. fMRI is fine for right now. I want to know, how can we fold in this multidimensional data around us? How can we get that data from participants?
Yeah.
We have so many new ways, actigraphy and EMAs, the momentary assessments, survey data, clinical data, behavioral data, neurocognition, on and on and on and on. And again, this is where I come back to ABCD and all of the data that is being collected for that study. How do you wrap all of that data into this package that somehow helps us understand these teeny tiny little fMRI time series?
Yeah. Yeah.
Right?
Yeah.
That's the true challenge.
Yeah. Yeah. And you're right. You're right. I mean, it's because it's immediately, unless you have something like your tools in this regard, you know, everyone is sort of like in their very narrow window, and that's not overwhelming. But when they quickly step back and they realize it's just overwhelming, you can't possibly get your head around that data without these sort of tools, as far as I'm
Right.
concerned.
So, I mean, that's why I don't think meta-analysis is going away.
No.
Right.
Yeah.
We will continue to publish papers trying to understand the brain, trying to capture brain behavior associations in reproducible, meaningful ways. And that will continue to lead to results that will need to be synthesized over time.
Yeah, I completely agree with you. And actually, it's interesting too, because, and I think it's a trend. And just to talk a little bit about the cultural shifts of younger students today, it seems that in general, you know, just going to OHBM, my impression is that at least half the people there do some form of meta-analysis, some form of at least tapping into some database. They don't really collect the data, but they more, you know, they collect the data from the literature and the other databases.
Yes, it's environmentally friendly. I've always said I was a pioneer in ecologically, sustainable fMRI research. I have a whole talk that I give to folks that's about being a responsible data analyst. I do think that it's absolutely brilliant that so many data are readily available. And for those of us who have started out in the earlier days and gone along that cultural shift, it is nothing short of transformative,
Yeah.
right? To have folks raising their fists saying, absolutely not, you'll take my data out of my cold, dead hands, to folks just being like, okay, and here you go. It used to be that you couldn't do fMRI unless you were at an institution that had the resources to invest in an MRI scanner.
Yes.
And so that limited who could participate in this endeavor. And so open access data, it's broadened access in our field. It's said, let's be more inclusive about who can play this game with us. And sort of counterpoint to that. So it's like, yes, open access, half the people at OHBM are analyzing freely available data. Wonderful, brilliant, fantastic. Teeny little asterisks that I say, it is super important that folks really take a second and think about the responsibilities that they bear when engaging with data that they didn't collect.
Yep.
Data that's open, you can click a button, open Neuro, boom, data set. It's right there on your computer and you can get to work. And it's important that people take a step back, take a pause, and really think about the data that they're working with, the research question that they have, and learn about the data acquisition piece as best as possible that they can.
Yeah.
A lot of my strong feelings on this really do come from my work as a site PI for the ABCD study. We have been collecting data for that study since 2016. These families are our families. They are important to us. Their safety and well-being means a lot to us. I am thrilled to participate in such a project that is getting into the hands of so many researchers and allowing them to analyze these very complicated, but really exciting data.
Yeah.
I love that ABCD data are being used so broadly. I hear we're approaching 1,000 studies based on ABCD data, and I think there's somewhere between 250 and 300 grants that have been funded by NIH to analyze ABCD data. And so I do think that when you're talking about these large complex data sets, if you're going to jump in, you have a responsibility to really learn about the data and make sure that the research question that you're asking is appropriate for the data that have already been collected. It sort of flips that traditional hypothesis testing design on its end, where you say, oh, I have a research question, let me design some data and go collect it and test my hypothesis. You have to say, the data are here, this is what they are, I need to really learn about at what time point and how old were the kids and what are the potential covariates that make sense here, and then move into the data. So I really stress, especially for students, to do their due diligence, to learn about the data before they jump in head first with their analyses, and as an absolute basic first step, to pre-register their analyses. You don't have to do the registered report way, but you can write down on a document what your analysis plan is, and then go put it on the Open Science Framework in a private repository so that you don't get scooped, because we don't want students to get scooped. But it gives you an opportunity to really map out what your usage of the data is going to be, circulated amongst your advisor, your co-authors, your collaborators, so that everybody is on the same page before you start investing the time to go down too far in a rabbit hole that may not be the appropriate pathway for your research question.
Yeah, no, I totally agree with that. And you stated really well how people, right, who don't collect the data, it is their responsibility to sort of be aware of that. And I always think it's still always a good idea I mean, there's something learned from collecting data and seeing what the artifacts look like and having a more visceral sense, but it may not be entirely necessary. It might be inefficient to require that in some sense. And there's advantages of that, but that's a different type of research that maybe people would go into than working on data collection or things like that. But yeah, and we'll definitely talk more about the ABCD in a second. But I think what you were talking about also is sort of like changing the incentives. With people collecting their data initially, it was sort of like all about their data, and their incentive was to promote, their lab, get research money, whatever. And if you can start, rewarding, realizing it's not a zero sum game, and rewarding people for, in some sense, for sharing, for contributing, for using that data set, which I think is happening slowly.
I really credit the NIH as being a key transformative influence in this respect. We had the Alzheimer's disease neuroimaging study, ADNI, that started collecting data to make available. But it wasn't until the call for applications for the Human Connectome Project that came out, where it was very crystal clear, this is a data collection project. You're going to collect the data and you're going to make it immediately available. And so it's very much like, if you don't like that, if that's not something that you see yourself doing, then don't apply to this RFP, right?
Yeah.
And so I just think that the HCP, we've learned so much from it. We've learned about the brain, but we've also learned about how we can do community-based science.
Yeah.
And I think it opened the doors to studies like ABCD, HBCD, UK Biobank. But without the HCP, we wouldn't have any of this.
Yeah. That I remember when that started as well. I mean, there was some debate as to whether it was premature. I think Mike Herda,
Yep.
yeah, he was, we were talking about it, and it was great. It was so forward looking to actually go ahead and just do it. And I wish they would keep on doing. I wish they would do a Connectome 2 or whatever.
Yeah, me too.
There's different versions of that, but that's gotten so much traction, that data set. It's just amazing, and it makes me think about the whole idea of, to try to get more control over the quality of the data collection. I mean, certainly with the UK Biobank, for instance, they have a certain scanner. They have a few centers around. You can almost imagine a model where you set up these sort of national centers that really have like a critical mass, and all they do is collect data a certain way, for in a well controlled way, because I really think that more can be done. There's a lot of room for curation of the data. Of course,
Yep.
it's important to try different things all the time, but at the
Yep.
same time, it's also important to have a certain way of collecting it that could benefit some regard.
So I really feel like I'm the first physicist in the history of people to say, yeah, technical differences, sure, but let's talk about the human factors here, right? Yes, we see a lot of variability due to different scanners, but we can model that. What we can't model are the humans and how they collect data, right?
Yep.
So if we are going to make the transition to ABCD, then
Yeah.
I will say this participating in this study has been nothing short of extraordinary. There were a lot of folks that reached out when it was announced that FIU was funded as a site, and they were like, hey, this is going to take over your life and it's bonkers. So are you sure you want to do this? We have this visionary team from UCSD. Terry and Sandy are our leaders in this study, and they have been so supportive and organized that I've just been in awe of their leadership. The way, and so swinging back to the way the data are collected, we had from the very first days we got detailed SOPs, standard operating procedures, that walk us through scripts, that walk us through procedures. They have done absolutely everything possible to make sure that data are collected the same at every single site. We have a site monitor who visits the site and grades us once a year. The site visit on our calendar is always an important event. There is a lot of focus devoted to cultivating our research assistants. They're the ones who are interacting with the families. We try to limit staff turnover as much as possible. These jobs are really designed sort of for that post-bac period where you get folks who they get their undergraduate degree. A lot of folks here at FIU get their undergraduate degree in psychology. They want to go to grad school, but they're looking for something to kind of bridge to that when they feel ready to move on to graduate school. So we got a lot of folks in the post-bac zone, and making sure that they are well trained, that they are comfortable, that they have the resources they need, that the support they need. That's my primary job.
Yeah.
I'm not interviewing families, but I need to make sure that they have what they need so that they can effectively manage the appointments and engage with families. Because at the end of the day, the most important thing about ABCD is retention.
Yeah.
The first 2 years, it was recruitment. Absolutely. First 2 years was a madhouse. I think I blacked out, months and months of it. But once we shifted from recruitment to retention, it came all about how we can engender this sort of flexibility so that we accommodate families if they're going through a rough time. So if you dig into the specifics of a specific person, participant in ABCD, and when they hit their appointment, and oh, by the way, there's this global pandemic, and so that suspended in-person assessments for quite a while.
Yeah.
There's so much variability in terms of having a very strict protocol, especially for timing of the different waves and what happens on the ground,
Yeah.
right? And at the end of the day, if I get somebody, and I was told that this is a good mindset by someone who was leading FIU, leading ABCD.
Yeah.
I was told that if I get somebody and I scan them at baseline and they ghost me for 9.9 years and they come back on the very last day that ABCD is operational and I scan them again, that's a successful time point.
Yeah. That's great.
Right? That's a successful data set.
Yeah.
With a study that's involving 12,000 kids, you're going to get a lot of variability in data quality, but that's the beauty of it,
Yeah.
right? It's big enough that there should be meaningful signal that we can still get even with all of this variability. But by planning the study and having such detailed protocols, and our RAs once a year, not only does ABCD come to us and evaluate our site, which gets everyone very nervous because we want to do very well, but also our RAs go to train the trainer every year. We're planning for it now. It's July of every year where they go to UCSD. So there's a lot of interaction at the site level to the consortium level for the RAs, and I really think that's just a major part of the secret sauce to ABCD.
So it's all the human, all this sort of like individual high quality interaction at the human level.
Absolutely.
Yeah.
Absolutely.
And So why don't we, I realize that most people probably already know exactly about the ABCD project, but why don't we just back up for a second? So it's the Adolescent Brain Cognitive Development Project. How did it get off the ground, and what is it? What are the goals of the project?
So it was Nora Volkov's. She's the mastermind.
Yeah.
She is the genius behind the curtain here.
Yes.
And it was I've just started serving on NIDA's council, which I think is interesting because I am not trained as a substance use researcher, right? I'm a data girl. But they keep reassuring me, no, this is what we want. My genesis into ABCD was from when the request for applications came out, because I have FIU does do a lot of adolescent substance use work, and so my colleague, my co-site PI Raul Gonzalez, I've had a number of different conversations with him over the years as he's trying to impart some of his wisdom on me. And so the way it was pitched to me is that with adolescent substance use, because this is such a incredible period of significant development in the brain, as well as sort of these maturational processes as adolescents discover who they are. And as the mother of 15-year-old twin boys, it is quite a rocky period. The idea that a single cross-sectional study could tell you about substance use, initiation, and escalation during adolescence, cross-sectional can't tell you anything. And longitudinal is so complicated because of all of the biological, sociological, environmental things that are going on in a youth's life. So really the only way that Nora would personally ever get answers that she was interested in is to go big or go home.
Yeah.
And to develop this humongous self-sustaining engine at this point, where you get enough participants and you go early enough in development prior to the initiation of a lot of substance use.
Okay.
And you follow them through. So we recruited participants when they were 9 to 10 years old. For many of them, this was before the onset of puberty.
Yep.
And we are following them for 10 years. So the study is going from 2016 to 2027. We were funded in 2015, which is when we first started working together. And so, it's this idea that NIDA could have continued to fund a lot of smaller, more affordable studies. And Dr. Volkov simply said, let's try something different.
Yeah.
And it's an inspirational story. I absolutely love it. I'm absolutely honored and privileged to be a part of the study. It's incredible to see it in action.
Yeah. No, it's right, and it's taking on a life of it. I mean, there's so many different groups, and it's sort of like this idea. I mean, I think like you were touching upon. It's the idea that there's certain questions that need to be asked in this sort of coherent, coordinated way, as opposed to, like, let's say, like, this is an interesting question. I mean, let's say Nora didn't do this, and then she funded a whole bunch of different studies, and then you tried, and then do you think you could come to similar conclusions with applying meta-analysis tools to those various different studies, try to coordinate them in some way versus this? I mean, how does this, and actually it not only answers a certain set of questions, but it opens up a really rich database for asking all kinds of other questions too.
Yeah, and so could meta-analysis have helped us without ABCD? Absolutely not. And here's why I feel this way. I just last month went to the ABCD Insights and Innovations meeting. First time that was offered. It was on your campus in Bethesda, and it was a gathering of folks who have been analyzing ABCD data and reporting results. And it included some ABCD consortium members, but a whole lot of folks who are not ABCD consortium members. And there was this moment of reflection at the end where we all kind of just took a step back and said, yeah, like the early days of ABCD were very scary. It was like, is this so big that it's inevitable that it is going to fail? Looking around at this meeting and seeing the incredible work that's emerging, no, this has been a resounding success. And I will have that goes on my tombstone, right? Like I helped do this, and that means something to me. But what I think right now is so critically important about ABCD is something that was viewed as a sort of a disadvantage or like a con in the beginning. And that is that the length of our protocol is, it's right there on the tippy balance of being a burden to participants. Folks, especially at baseline when these kids were 9 and 10 years old, they're spending a half a day, a full day, hours and hours with all of the different assessments.
Wow.
If you go onto the ABCD website and you look at all of the different protocols, physical health and mental health and neurocognition and culture and environment, and it's just domain after domain after domain, and that's just the youth. Then the parents go through the protocol. Then there's teacher surveys. Like, the wealth of information, when we first started collecting it, we were like, this has to be too much. This is not sustainable. But now, getting to now, researchers are really starting to come into their own in collectively leveraging this very rich data set. Shifting in the field is also, there's a lot more recognition and awareness that, again, these brains are not operating in a vacuum. And if you are not taking into account the structural and social determinants of health, then what are you even doing here, right? ABCD has captured a lot of socio-environmental variables that haven't been captured in previous studies. And I do think it's because a consortium-based approach was used, so that you had the epi folks who were like, hey, by the way, you really need to get this and this. Neuroimaging has historically been led by folks like you and me, physicists and engineers. And we're like, just get the brain in the scanner. That's good enough, right?
So to see something happen, and exactly.
Nailed it. I've got the secrets to the universe now. And then another important thing, and again, this has been progress from the HCP data set, and that's ABCD in its multi-site approach has been able to capture a diverse demography for the first time in our field. There was a special phase towards the end of recruitment where prioritization was made to over-enroll low-income families and families from racially and ethnic minority backgrounds. And so as a result, you do have a much more diverse data set in terms of who people are in ABCD than in other data. And I think that has really been key, because that, combined with our structural determinants of health data, has allowed us to ask really meaningful questions that I saw at this recent AIM meeting about childhood poverty, about gender diversity. Oh my goodness, Alexi Potter at University of Vermont, she blows me away with how critically important her work is within the lens of these statewide mandates about gender diversity right now. So neighborhood disadvantage, and then just having a deeper viewpoint into folks and their lived experiences and how that influences neurodevelopment during adolescence. I get really excited. I get really fangirly for all of the folks who are using these data in truly remarkable ways. I think it is a great time to be a neuroimager.
That's awesome. So, is there anything from the ABCD project that, you were talking about sort of there have been many papers already published, but is there anything that sort of was uniquely surprising that came out so far, or is there anything that was unanticipated that's an issue, or is there, there's anything that basically took you by surprise as far as so far?
Yeah, I'm not a take me by surprise kind of girl. I feel like, things are what they are. But I gotta say that I don't think anybody can answer that question without pointing to the Merrick reproducible study that came out. What was that? 2021, 2022.
Yeah.
Thousands of participants are needed for brain behavior associations, right? And so collectively, our field gasped and clutched their pearls, and there was a lot of hand wringing about what are we going to do, and is neuroimaging dead? And I love those discussions because they have meaning, and our we need to embrace our initial gut reactions, and also embrace the process that means that now we're going to have some real meaningful conversations about, okay, what do we do next, and how do we need to shift things? We're certainly not going to take all of our MRI scanners and toss them in the bin and move on to the next shiny thing. And so it's been nice since that paper came out to really see, science moves slow, right?
Yeah.
And it should. I love slow science, slow, thoughtful, deep science. But I've seen a very quick shift to embracing more multivariate methods,
Yeah.
partial least squares correlation,
Yeah.
right? So
Yeah.
I like that paper was basically a shot to wake us all up. And now we're all kind of saying, okay, well, what are we going to do? Let's stop doing the same old thing. Let's shift our strategies a little
Yeah.
bit.
That was a wonderful paper. And
Mhm.
there's a lot included in it in the sense that, it may be said, well, maybe each individual. And so my intuition, at least, is that, and it sort of helped catalyze the sort of the area of study of individual variation, because there's like, well, maybe there's so much heterogeneity even within psychosis categories, or heterogeneity of brain morphology, that, when you try to put it into a template, a lot of the parcels are, everything's mixed and you get the strongest effects, which are real, but there might be better ways of accounting for individual variability. And we first have to understand that a little bit more. So catalyze that direction. It also catalyzed, I feel, and this is an interesting topic in itself, the idea of dense sampling.
So I do have tremendous respect for Scott Merrick and all of the, Brendan, all of the authors on that paper. I think I love how they said, let's make this a punch in the gut. And they took very, aggressive has a negative connotation, but they took very strong language with the conclusions that I think is what set a lot of people off, right?
Yeah.
And I think that was beautiful because they started a conversation. And I think what we are learning is that brain is so complex, we do need a lot of different approaches to try and uncover all of the dimensions. So yes, I think dense sampling is critically important and can tell us so many different things. And I'm happy to talk about Russ's pioneering work in this area with my connectome, but I will absolutely go fangirl on you with Emily Jacobs' work in this field. I think that her input on understanding how women experience functional brain connectivity through their monthly cycle is very, very important.
Yeah.
But I also want to tap into what you said, with all of these data, in thinking about what the future holds. I'm not really interested in a new imaging modality. I think we need new data frameworks that help us capture this multidimensional data. We've made such progress with ML AI. And I don't want to be one of those university administrators who's like, what we need? We need some AI to solve this problem. But what we need is some AI to solve this problem, right? We have so much data, and until we develop an analytic framework that will help us fold everything all into one nice little mixing bowl and find some structure from it, then I think that we'll continue to struggle.
Yeah, I completely agree. And I think at least emphasize the fact that the field is maturing in many different ways. I mean, it's like
It is. I love that, right?
Yeah, and it's a slow process. I mean, it's like, people, like you said, it's sort of overall, you can sense that, people are getting more sophisticated about how they collect the data, how they interpret the data, how they pool the data, how they curate it. And it makes me, I still, the ultimate goal, and I keep on thinking of these ultimate goals, but maybe it's premature to even do that. But I would like, it would be great. I mean, the ABCD study is wonderful in that regard, for reaching a deeper understanding of brain behavior and correlates, and also signatures of disorders and development. But I'm still waiting for, people are still waiting for fMRI to sort of really get clinical traction. It can be used for pre-surgical mapping. That's great, but it's nothing that gets vendors' attention or people, in terms of a market for clinical application. And so what do you think? Like for instance, how would you get from, like, for instance, an ABCD study, or in general, what we're doing now, to how would it be used clinically? Like, put a person in a scanner for trying to make a diagnosis.
I feel like we're just at an infancy stage,
Yeah.
and even wrapping our mind around that. When you talk to non-fMRI folks in the clinical world, there's this what has it done for us? What are you doing for us? Right? It's like, okay, dude, I get it. Like,
Yep.
I understand. We're still not helping you in very practical ways. And the answer that I always come back to is that we're still trying to wrap our minds and our data around heterogeneity.
Yes.
And so you take conditions like ADHD and autism, and I'll go to the neurodevelopmental disorders just because I know them a little bit more. There's so much heterogeneity in symptom presentation, in response to treatment, in everything, that's where fMRI can lend a little bit of input at this point. And that's very much a research bent. That's not something that a clinician will say, oh, thank God you solved it, right? But if we don't do this bit first, then we're not going to get to the point where maybe fMRI can help us understand how to tailor treatment to an individual.
Yes.
Heterogeneity, if we start carving that out, we can start developing profiles of individuals, and we can start saying, okay, here's what ADHD actually is. It's these 3 categories or 12 categories.
Yes.
And then you maybe fit in here. And then ultimately, maybe in the future, that means that if we did this behavioral therapy, then that would help.
Yeah, I totally agree with you. And I think that heterogeneity, and what I tell people actually, because sometimes they compare fMRI to, like, for instance, spectroscopy. Spectroscopy, people joke about that, where, MR spectroscopy, at least, where
Yeah.
it's always the next.
I did that too.
It's always the next thing that's going to happen. It hasn't happened.
Yes. I was told that in the mid 90s. Oh, it's right on the cusp.
Right.
It's coming. Like, oh.
And it's great. But I think that there's a fundamental difference. And I just, in some sense, I tell people all the time that with fMRI, it's not a matter of sensitivity. fMRI is plenty sensitive.
Yes.
It's a matter of, it's sensitive, but it's a matter, like you said, of the heterogeneity, of trying to get a sense of the variability.
It's the humans.
Yeah.
It's the humans. I feel like the tech is fine for right now. Although I love that people are developing a tech, that's just not what I'm drawn to. I'm drawn to trying to figure out these crazy humans.
Yeah.
I love them.
Yeah, exactly. And so in that sense, it's a great problem to have, because we're realizing,
Yes.
wait a second, this is more interesting than anything we imagined, and let's try to get our heads around it. And we have this great tool that's sensitive enough to do this. So.
And I firmly believe that collaborative science is the best science. I've never wanted to be a scientist who's locked away in a dark room, working completely on my own, waiting for this, lightning insight to strike. I like interacting with folks who have different backgrounds than I do. And so I've spent the last couple of years, I'm here in Miami. It is such a diverse city. We have a lot of Latino immigrants. We have folks who have come here from Central America, from South America, from the Caribbean. Miami is the most amazing city, ever. And what I like is that I've spent a lot of time learning from folks in my institution about work that's being done to address Hispanic health disparities. And I've spent a whole lot of time learning from social and cultural psychologists about the work that they do and the tools that they have. And I very much feel in participating in the ABCD study that study was made better because it included such diverse viewpoints and folks from all different walks of life. I feel that we have an opportunity to be collaborative, and that we really need to embrace working with someone that just comes in from a completely different perspective, because that's where true innovation happens, right at that intersection.
I totally agree with you. And I think that the more diversity, the more collaboration, the better, especially with these areas that you just can't be an expert in every aspect of what you're doing.
Yes, exactly. Yes, I collaborate because I'm lazy. No. Like, no one can learn it all, right?
Yeah, and I think actually the real trick, and I think this could be actually an interesting meta tool in some regard. That the real trick that I found in collaborating is that it's hard. It's the mechanisms, the mechanics of collaborating. Like, how do you collaborate? I mean, you usually say, well, I'm going to do this part, and I'm going to do this part, that we're going to talk about it, and then how do you? It's really, there's this coordination that sometimes works and sometimes doesn't. It depends on personalities, depends on sources, and having a tool that sort of fosters collaboration in some regard could be interesting. sort of.
Yeah, I love that. Again, I'm very much swayed by my location on this planet, and Miami is very much in the U.S. The front line of climate change. And our institution has a lot of amazing researchers, ecologists, environmental resilience experts, and I've been spending a lot of time talking to them. And so for me, collaboration, sort of the linchpin is communication.
Yes.
Like, and not just like, can you sit down and have a conversation with folks, but can you get into what your data look like and what their data look like? Are there ways in which the climate folks data have structures and information that we can use to interface with the brain imaging data to look at the effects of climate change on the brain, for example,
Yeah.
right?
Yes.
And so I feel like, just getting in a room with folks and really kind of interrogating them, drilling down on what their data look like, how they collect it, especially these environmental folks who are, out there in wastewater facilities and dealing with septic tank
Oh my
overflows and
gosh.
all kinds of really, really terrible things.
Yeah.
But really learning about what they learn when they find a puddle on the ground that tells them about, if they look at how much caffeine is in that puddle and how much sucralose is in that puddle.
Huh.
Those chemicals only come from one pathway to be out here on the sidewalk.
Yeah.
That's wastewater contamination. And they have different half-lives for decomposition. And so that tells you long-term and short-term effects of exposure to these environmental contaminants.
Wow.
So yeah, so like that's the cool stuff, is when you really start drilling down into what data are people collecting that we could then use to fold into a truly transdisciplinary study to look at something that people really should be caring about.
Yeah. Yeah. That's so interesting. To communicate, but also to coordinate, like, each person's goals and agendas
Yeah,
and
right. Mutually beneficial.
yeah. Yeah. But that's really interesting. I mean, there's so many questions to be asked with that regard. So it seems like, right. I mean, you paint this picture, and I think you're sort of right at the front lines of pioneering this with a dozen or more groups at the very most. I mean, you're definitely leading the way as far as, trying to do this sort of thing, trying to foster sort of the further cultural shift in this regard, because there's questions waiting to be asked only with these sort of things, these sort of tools and collaborations. So okay, one practical question that was sort of burning in my head, and I do, and I sort of I recently talked to Peter Fox about this, and I think it's an interesting question with fMRI. I mean, certainly fMRI data, the way we're looking at it now, and certainly it will change and modify, but as you go to higher resolution, so it seems that describing areas either by coordinate systems or even unthreshold maps, if they're not perfectly registered, is a problem if you're doing, for instance, layer fMRI. So I'm trying to grow the area of doing, layer specific cortical layer fMRI, where
Yeah.
and that's actually, I think a lot about, okay, how do you pool data like that? It's really specifically one subject, and you try to, normalize relative to individual, cell sign gyri, but and then landmarks and things like that. But I think people are working on it. But it's a tough problem. So those are problems that I think that put that into a database where you can start to make detailed models of fMRI activation is hard. I think it's a challenge.
So what I like about meta-analysis is its flexibility,
Yeah.
right? The specific tool isn't necessarily what matters. The general approach is what matters. It's a tool that allows us to step back and reflect on what we know.
Yep.
And it's a simplistic tool, right? Coordinate-based meta-analysis is, I mean, you are literally taking the centers of mass of the coordinates and just saying, okay, where do they overlap, right?
Yep.
Of course, there's some statistical nuance there, but that's the main point.
Yeah.
And so as long as you're using an imaging modality or a technique that's telling you something about localization of brain function, then ultimately I'm confident that we're going to find a meta-analytic approach that's going to help you, right? Yeah. And so yes, there are some hard registration problems, and the algorithms are definitely going to need to evolve to accommodate layer fMRI, but I don't see any of those as being unsurmountable challenges, right? If you do a deep dive into early studies and look at how people used to talk about the template that they used for spatial normalization, or not even talk about it, or say things that are objectively wrong, we used SPM 96 to normalize our brains to Taylor-Ack and Chernow space,
Yeah.
right? Like, no, you didn't. And so in the early days of meta-analysis, I'm sure there would have been a lot of researchers who would have really dug into the template issue and gone, absolutely not. This is chaotic garbage. I'm not doing this. Let me go do something else.
Yeah.
And I just kept saying, it'll be fine. It'll be fine. Right? It's a little messy, but don't worry. Just put it in. It'll be fine.
Yeah.
So, every time you do a meta-analysis, it's coordinate based. You do have to tag that particular set of coordinates with a template.
Yeah.
I am sure that those are tagged wrong most of the time.
Yeah.
Right? And yet we are still getting meta-analysis results that align with new studies that are telling us information that looks important and meaningful.
Yeah.
So I feel like the margin of error on this is so much wider than we think that it is. And so if you ever did want to start jumping into meta-analysis of layer fMRI, then jump in, right? See what you get.
Yeah. Yeah.
Because I bet the margin of error is wider than that it can tolerate is a lot wider than you think.
Yeah. And I think I definitely agree. I think it's solvable. And I think actually in the process of solving it, we'll probably gain more insight in terms of, what the variability of the brain is, how it's organized, how it's the structural variability too. So
yeah, it is very cool stuff. I love it. Thank you.
well, I mean, yeah, either way, it's a lot of fun, but yeah, we're trying to figure out how to make it more useful as well. But we covered a lot, and we covered a lot of really good. I'm glad we had a chance to discuss at least a little bit the ABCD project. I actually never had a guest on talking about it. Maybe I should have a whole podcast just devoted to that.
Oh, you could absolutely.
Yeah.
Yeah. I'll tell you who to ask. We've got some amazing, and I love that, we've got some folks that are like core OHBMers, and then we got a lot of folks that are, the developmental cognitive neuroscience community, and then we have a lot of other people from outside both of those communities. So I do love that it is the confluence of a lot of different domains.
Yeah, that's very cool. That's very cool, and I can't believe it's already been almost a decade, or just
wild.
Yeah, that's amazing. So, all right, well, I think with that, well, actually, one last thing I usually have. So, do you have any advice for starting out researchers? What sort of tools, or obviously it depends on what they want to get into, but if you're getting into fMRI or in general, what do you think is important to learn?
I always come back to, like, if I had to pick one thing, it's pre-registration.
Yeah.
And it's not because it's a research tool, because I think there are a lot of research tools that everybody. I think Learn Python, it would probably be my number two, but my first is pre-registration, because it is such a communication tool. When you're first getting into research, it really is about developing a relationship with your advisor. And if you are having conversations where one of you is talking about topic A and the other is talking about topic B, and you don't realize that A is not equal to B, then downstream, there's going to be a train wreck. Pre-registration levels that playing field. You put your ideas onto paper, your advisor takes a look, adds their input, and it just really makes things more transparent. Yes, for the community, and that's great, but more about those baby steps when it's about you and your advisor.
Yeah.
And then the other thing, again, my advice is more human than tech, is that not to be scared of doing things differently to forge your own novel career path. There's not a whole lot of folks out there that are like me, that were trained in a true physics department, that are doing cognitive neuroscience research. I didn't realize when I started on this crazy ride that there were so many folks that I would end up being, faculty and collaborators with, that came from academic backgrounds. And so as someone who comes from a non-academic, non-science background, I struggled a bit in the beginning, and I was a little bit slower to kind of figure out the system, because there is a system and it is a secretive system. But I also didn't have any preconceived notions about what I needed my career to look like. And so having a faculty position in my early days at a medical school, it gave me the structure that I needed, and it helped teach me how to be a scientist in a way that I still embrace today. But when that medical school environment got too rigid and I needed a platform to move in a new direction, I moved to a College of Arts and Sciences, and that gave me more freedom intellectually and almost spiritually, right? But if I would have gone directly down that path, I think I would have floundered because it is a looser environment. And so I just feel like, everybody has their own unique career path, and that's okay. Right? Embrace that. It's not a disadvantage, it's an advantage of what our opportunities are.
Yeah, that's really great advice. I mean, I think everyone is sort of fearful if they're not doing the right, if they're doing the right thing or not. It's like, don't worry, just
ask good science questions.
Yes. Yeah, exactly. And right, like you said, work with your advisor, have pre-registration, but then be creative, do whatever. And yeah. All right. Well, thank you so much. This has been a really really fun conversation.
You're welcome. This has been a lot of fun. Thank you.
All right. Thanks.
Neurosalience is brought to you by Organization for Human Brain Mapping. This week's episode is produced by Xiqian Michelle Li and Ömer Faruk Gülban.